everyone. In today's class, we are going to study structure of contractile protein, that is part 5 for locomotion and movement. Now, if we talk about the previous classes, we have studied about the structure of skeletal muscle. We have also studied about the muscle fiber structure. When we talk about the skeletal muscle fiber has certain amount of protein, certain type of protein. The only two protein we have studied in the previous classes is actin and myosin, but only actin and myosin is responsible for the contraction. Let's figure it out in today's class. Skeletal muscle fiber consists of two types of protein. One is the contractile protein, second one is the regulatory protein. The word itself says that the contractile protein, that means what? It helps in the muscle contraction. It generates the force that helps in the muscle contraction. Suppose if I want to lift my hand, so that is what will happen? Muscle contraction will happen. But am I not supposed to bring back my hand back, or I'll be just raising my hand for hours and hours? No, I have to come to the relaxation position. So this contractile protein helps in the muscle contraction, but some other protein has to be present which should regulate the switch on and switch off of this contractile protein. This type of protein we call it as regulatory protein. That means this protein regulates. What do you mean by regulates? Whether switch on of the contraction or switch off for the contraction. Switch off contraction means relaxation position of the muscle. Okay. Now, where the switch on and switch off happen? During the interaction between actin and myosin. If you remember my previous class, what did I say? That the actin is going to slide over the myosin. So when it is sliding over the myosin, जादू थोड़ी है कि अपने आप से बोल दिया slide कर दो. No, there will be the binding, there will be the interaction between myosin and actin. That interaction should be stopped when we talk about the relaxation. So who will stop this? And who will start this? Of course, who is going to regulate? Your regulatory protein. Now, very important with respect to the NEET, what are the examples of contractile protein? PYQ. So, what are the two contractile protein? Actin and myosin. Myosin more to thick filament, actin is a thin filament. Regulatory protein, we have two types of regulatory protein. One is the tropomyosin and the second one is the troponin. Tropomyosin is attached to the thin filament and troponin is found on the thin filament. When we'll study about the structure in detail, then you will understand. So let's go with the structure of thin filament, then we'll follow the structure of the thick filament. When we talk about the thin filament, till now we have studied thin filament as actin, but is thin filament consist of only actin? No, it is composed of three different protein. Are the three different proteins are contractile protein? No, it's a combination of contractile and the regulatory protein. So first protein we are going to talk about is the actin, and as we have studied, this is the contractile protein. That means it is going to help in the muscle contraction. So here you see the structure of the actin. This actin which is globular, we call it as G actin because कैसा है? Globular actin है. This is globular and this is monomer. That means many G actin will combine together to form a filamentous actin, which is a polymer. So basically actin is a polymer which is made up of many monomeric unit. What is the monomeric unit? G actin is the monomeric unit. This is inactive state, that's why it has to get to the polymerized state and then it is soluble in nature, right? So one actin, if I'll talk about, it is G actin, which is globular. It has a site that is called as active site of the actin. What do you mean by active site of the actin? This is nothing but the myosin binding site. So don't get confused. At some places in the NCERT, they wrote active site for the actin. At some places, they wrote myosin binding site. Both are same. Active site क्यों बोला? Because some reaction will happen over here. Myosin will come and bind. Myosin binding site क्यों बोला? Because myosin will bind to the actin only and only on this side. Nowhere else myosin can come and bind. Okay, so this G actin will polymerize. That means many G actin will combine together in the presence of magnesium to form a filamentous actin. We call it as F actin. How is this actin? This we call it as polymer. ये कैसे थे? Monomer थे, single unit थे. Now these are active and insoluble. These filamentous will form a double helical structure. So ये एक filamentous chain है and there is second filamentous chain. So how many filamentous chain will be present? Two filamentous chain will be present. So it form a double helical chain structure. So your actin occurs in two form. One is the G actin, that is monomeric unit, and then another one is the F actin. It has which site? Myosin binding site, which is also called it as active site for the actin. Now it is made up of 2F actin. So if I'll ask you about the actin, actin is nothing but 2F actin. You will not say it is 2G actin. See, basically, if you want to make a Maggi, you should have a Maggi packet, you should have a masala packet, right? So same way, my actin is 2F actin, 2 filamentous actin, which is made from many glomulars, many G actin. ठीक है, in the presence of magnesium. This process we call it as polymerization. Now let's come to the next one. So what is F actin? F actin is nothing but the helically wounded to each other to form a double helix. So 2F actin is helically going to be wounded to each other to form a double helical structure. ठीक है. Then next protein which is present in the thin filament is your tropomyosin. Is it also a contractile protein? No, it is a regulatory protein. That means it decide whether the contractile protein has to be switched on or switched off. This is a fibrous protein and it is a filamentous structure. When I say filamentous structure, again like this, it will be present. Okay? It runs close to the F actin. See this pink color one is the F actin and the blue color one is the tropomyosin. Can you see it runs exactly closer to the your F actin? Now why does it runs exactly closer to the F actin? Because it has to cover what? Myosin binding site. If the myosin binding site is not covered, what will come and attach to it? Myosin, and automatically contraction will take place. We don't want, just imagine that I don't want to raise my hand and suddenly the hand raised will be there. You know, that is because of the what? Because the myosin binding site is not covered. Anytime myosin can come and perform the contraction of the muscle, right? So this tropomyosin, what is the function of this? It is going to cover the active site of the actin, that is myosin binding site, in order to prevent the formation of cross bridge. This cross bridge you will not understand now. You will understand when I'll explain you the mechanism. Now, when this process will happen, when this covering will happen, when there is no contraction, right? Then let's jump to the third protein which is involved in the thin filament formation, that is your troponin. Again, is this a contractile protein? No, it is also a regulatory protein. It is a complex trimeric. See, both actin and the tropomyosin are the filamentous protein, but this is trimeric protein. That means how many units are present? 3 units will be present. It is distributed at a regular interval where on the tropomyosin. So this is your, see, this one I have tried to show the filamentous actin. Okay, the filamentous actin, and this green color is the myosin binding site. Then on myosin binding site, what is present? Tropomyosin is present. This blue color one. यही चीज दिखाया. ठीक है, let me erase all these things so that. Oh. Now, on this tropomyosin, this blue color one, what is present? What is present? Your troponin is present. What is present? Your troponin is present. Troponin has how many parts? 3 parts. So overall, if I'll ask you what is the function of troponin, during the resting stage, a subunit of troponin, mind it, a subunit likha hai, that means only one single subunit, that I'll tell you. It masks the active site of the myosin so that actin filament will not be able to bind with the myosin. ठीक है ना? Now see here. What are the 3 subunit? You can remember it as TIC, TIC, or CIT, SIT, whatever way you want to remember, you can. ठीक है? नाम में ही है. TP means troponin. Troponin T means tropomyosin. Tropomyosin is going to bind. My bad. T is for the tropomyosin. So TPT, that means this subunit of troponin is going to bind to what? It is going to bind to the tropomyosin. So if this is a tropomyosin, the subunit which is binding to the tropomyosin is the TPT. ठीक है? Then TPI. I means inhibitory. What I should inhibit? I should inhibit myosin binding to the active site of the actin, that is myosin binding site. So on this side, that is what is present? TPI. TPI is the inhibitory protein which masks the active site of the actin. This is PYQ. Which subunit of the troponin masks the active site of the actin? That is nothing but the TPI, inhibitory protein. ठीक है? Now this inhibit actin myosin interaction because it is not going to expose the myosin binding site. Then TPC, naam mein hi hai C, that means it binds to the calcium. During muscle contraction, it will bind to the calcium. This shifts the troponin and tropomyosin from the active site of the actin, which causes the muscle contraction. This you will not understand now. You will understand this in the mechanism. अभी के लिए बस इतना समझ लो. एक एग्जांपल ले रही हूँ. This is your, मान के चलो, G actin. On this, myosin binding site is there. On this, what is this? One second. हाँ, on this you have a troponin. ठीक है. अब this troponin पे what is present? Your one subunit, that is TPT, is present. Then another subunit, that is TPI, will be covering the active site, that is masking the active site, that is TPI. ठीक है. TPI. Better if we can write, this one as a TPI and this one as a TPT. Sounds better. Okay. Then another one is going to be present over here, which will bind to the calcium, which is released from the sarcoplasmic reticulum. Do you remember in the first class we have studied, second class we have studied that the sarcoplasmic reticulum is going to store the calcium. This calcium will come and bind to this TPC, and then automatically this whole subunit will be moved. When this is moved, what will happen? The unmasking, that is TPI, is going to vacate this position of the active site, and then it will also move along with the tropomyosin. So what will happen? The active site will be vacant. Okay. Tropomyosin will be shifted and the whole trimeric unit will be shifted, and the myosin will be able to come and bind to the active site. Right. This is the whole mechanism. You will understand this in the upcoming classes. So this was whole about the thin filament, sorry. Then let's jump on the structure of the thick filament. What is the structure of thick filament? We are going to study about the myosin. See, thin filament is made up of three things, that is your actin along with the tropomyosin along with the troponin. Troponin consists of three subunits. ठीक है. But यहाँ पे uh, myosin filament, thick filament mein only we are going to study about the myosin. What is myosin? Is it a regulatory protein or the contractile protein? It is a contractile protein. This is again a polymerized protein. How is the actin? Actin is also a polymerized protein, that is many monomeric unit, many G actin will form together, bind together in the presence of magnesium to form what? Your actin, that is filamentous actin. So वैसे यहाँ पे myosin is also a polymerized protein. That means इसका भी कोई monomeric unit होगा. इसका actin का monomeric unit what it was? It was G actin, right? इसका monomeric unit क्या होता है? Meromycin. What is the monomeric unit for the myosin? It is meromycin. Many meromycin will bound together like this to form what? This is the wrong way to say. Okay, many meromycin will bound together to form what? Myosin. That is one thick filament. Okay, now let's understand this. Each meromycin, मैंने क्या बताया? Let's imagine one meromycin, one meromycin, one meromycin, one meromycin. Many meromycin will combine together to form what? Myosin. Right? Now let's understand what does one meromycin comprises of. Okay, each meromycin have the two important part. What are the two important part? One is the heavy chain, other one is the light chain. I'll show you this in the diagram. First understand. Okay, this heavy chain, why do we call it as heavy meromycin? Because it has the high molecular weight. Why do we call it as light meromycin? Because it has the less molecular weight. Okay, now what is this light meromycin called it as? It is the tail part of the meromycin. We call it as light meromycin. Now how many tail part will be present? Two units will be present, and this is fibrous in nature. Then heavy meromycin consists of two components. What are the two components? Head and the short arm. How many short arm will be there? Two short arm will be there. How many heads will be there? Two heads will be there. So two units. How is the head? तुम्हारा हेड कैसा है? ग्लोबुलर है, सो इसका भी ग्लोबुलर है. What is present over here? Very 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 important. Actin के पास क्या था? Myosin binding site था. अब बाइंडिंग चाहिए तो एनर्जी चाहिए. एनर्जी कहाँ पे यूटिलाइज होगी? कैसे? एनर्जी किससे मिल रहा है? एटीपी से. यू गेट एनर्जी आफ्टर ब्रेकिंग द एटीपी. सो एटीपी कहाँ पे टूटेगा? हेड में. it has ATPase enzyme which breaks the ATP. So it has two sites, that is the binding site for ATP and active site for actin. अभी ये diagram से समझो तुम्हें सब समझ आएगा. So this is the diagram for the meromycin. This whole is one meromycin. Now if you will see, this is the globular head. How many globular head is present? One globular head, and this is the another globular head. तो कितने हैं? Two हो गए. Then this one, let me change the color one second. This one we call it as short arm. How many short arm? This is one short arm, and this is the another short arm. So how many short arms are there? Two short arms are there. Then tail देखो, how is the tail? It is fibrous in nature. How is the tail? It is fibrous in nature. कितनी है? This is one tail and this is another tail. So how many tails will be present? Two tails. This tail part we call it as light meromycin part, and this head along with the short arm we call it as heavy meromycin. Heavy meromycin. This head along with the short arm forms the cross arm. This is PYQ. Tail and short arm will not form the cross arm. Who will form the cross arm? That is head, and the short arm will form a cross arm. Now, very important, what is present in the head? You can see यहाँ पे what is present in the head. There is actin binding site. देखो यहाँ पे this is actin. यहाँ पे myosin binding site था. That is myosin is going to come and attach, right? So same way myosin पे actin binding site रहेगा. That means it is going to go and attach to the myosin. ठीक है, this part is not going to go and attach to the myosin, sorry, actin. Only and only it is going to attach with this part. And what is this part? This is actin binding site. Okay. Then the another site which is present over here is the ATP binding site. So here ATP will come, attach, and the ATP will be broken down. ठीक है, ATP binding site पे क्या है तुम्हारा head में? What is ATP will bind over there. Head में और क्या चीज है तुम्हारे? ATPase enzyme activity है. That means ATP binding site पे ATP bind तो कर गया, but I have to break this ATP, then only you will get the energy. So वो कौन करेगा? ATPase enzyme करेगा. वही चीज यहाँ पे समझाई है तुम्हें. So it act as a head, act as a ATPase enzyme that breaks the ATP. And what does it have? Binding site for ATP and active site for the actin. ठीक है, देन क्रॉस आर्म की बात करें तो हेड एंड द शॉर्ट आर्म प्रोजेक्ट आउटवर्ड. कैन यू सी इट इज प्रोजेक्टिंग आउटवर्ड? इट इज प्रोजेक्टिंग आउटवर्ड एट द रेगुलर डिस्टेंस. पहले एक आएगा, फिर लेट मी शो यू इन द, लेट मी जस्ट रीड दिस वंस. रेगुलर डिस्टेंस एंड द एंगल फ्रॉम ईच अदर, my bad, head and the short arm project outwards at the regular distance and a angle from each other from the surface of the polymerized myosin filament. Let me show you over here. See this. Can you see? This is one head and the short arm, one head and the short arm, one head and the short arm. That is one cross bridge, one not cross bridge, cross arm, one another cross arm. This is one cross arm, another cross arm, another cross arm. So can you see it is at the regular distance and projected outward? This is what is written over there. Okay. Now magnesium helps stabilize, this is an extra information. Magnesium helps stabilize the interaction between myosin molecule and the actin molecule. So दोनों के लिए ही, see this is one meromycin and many meromycin. Oh, I have drawn over here. So this, see here. So this is going to form a myosin. Many meromycins are going to join together to form a myosin. इसके लिए भी what is required? Magnesium. Co-factor for ATP hydrolysis, that is ATP को break करने के लिए भी what do we want? We want magnesium. That's all about the structure of your contractile protein. Thank you.